Okay. Oh, no, back here. So this first part just talks more about what makes up blood. And while I'm here, I just want to point out a few things. In this kind of material, and any textbook is going to provide this sort of thing, the numbers I'm not crazy about. For instance, I said this earlier, they say water is 92% of the plasma. I think it's a little too variable of a number to be that precise. So I'd prefer to work with 90%. Of course, if this becomes 90%, then the plasma proteins, which is 7, and the solutes, which are 1 some odd, doesn't add up to 100. So you'd have to adjust those numbers. But just think of this as being rounded off to 90, because it can vary from person to person, and for one person, quite a bit. Okay. And it explains what the different things do. The plasma proteins, there are 3 types. I mentioned albumin. And it's primarily there to make blood thicker. It's to make it a more hypertonic solution so that it's isotonic with other solutions in the body. There are globulins, of which there are several types. We're going to talk about the immunoglobulins, or antibodies, when we get to the immune system, and we won't really talk about the other globulins. And then fibrinogen is a blood clotting factor, which I'll talk about when I get to blood clotting. And then they have regulatory proteins and other solutes, so hormones and enzymes, nutrients, gases, wastes. It's all listed here. And then for the formed elements, which are the cells, we got erythrocytes, which are red blood cells, 99%. Leukocytes, or white blood cells, are less than 1%. And then weird little formatting issue here. In this box, it says leukocytes less than 1%, platelets less than 1%. Then down here it says platelets less than 1%. Weird quirk, they put platelets less than 1% twice. So just ignore it here because it has its own row there. We'll talk about the white blood cells in the immune system, and the different types of them and all that sort of thing. Platelets are about blood clotting, so when I talk about blood clotting, I'll include that stuff there. The next section is about the production of these types of cells, which takes place in the red bone marrow. And the process is called hemopoiesis. If we're going to talk about that production of blood cells, if we're going to talk about a particular type of cell, the hemo part of this word can be changed out with a specific root. So making red blood cells is erythropoiesis for erythrocytes. Making white blood cells is leukopoiesis for leukocytes. And making platelets is thrombopoiesis. A thrombocyte is a platelet. This flow chart just shows how the stem cell in the red bone marrow gives rise to different types of blood cells through a few stages of intermediate stem cells. This first little thing where this one cell divides into 2 and one of them cycles back, that's just that the hemocytoblast, the stem cell itself, every time it initiates blood cell production, it divides, and one of its offspring cell remains as that pluripotent stem cell. But the other one then differentiates into one of 2 intermediate cell type stem cells. This one over here, called a myeloid stem cell, gives rise to all of the blood cell types except for lymphocytes. Lymphocytes come from the lymphoid stem cell. And these are the main cells of the immune response, which we'll talk about in that chapter. But the other 4 types of white blood cells, which also have a lot to do with immune response, and red blood cells and platelets all come from the myeloid stem cell. This big cell here that kind of looks like a paint splatter, it breaks up into little pieces, which are the platelets. Okay, so this big cell is made and it breaks into platelets. It's just that sort of extra step there. Actually, let me make this picture bigger. Okay, so here's that big cell. It breaks up into the platelets or fragments. This is a red blood cell. And if you look at it closely, and the picture itself isn't really highlighting this, but it doesn't have a nucleus. Okay? There's a nucleus in the precursor to the platelets, and there are nuclei in the white blood cells, but there's not a nucleus in the red blood cell. There is a nucleus in the precursor to the red blood cell, which we can see kind of breaking down here, and a nucleus in its stem cell. But once it's a mature red blood cell, it doesn't have a nucleus. Actually, red blood cells don't have anything in them except for hemoglobin. Okay? They don't have organelles. They do have a cytoskeleton to keep their shape, but they're really just bags of hemoglobin that carry oxygen. Okay? Now, any cell in your body has to make ATP and drive some energy-dependent processes. And that's true with red blood cells also. Where do we normally make a lot of ATP in any cell? No. Yeah, mitochondria make ATP. Red blood cells do not have mitochondria. They don't have any organelles. They can make ATP because in any cell, the first step in making ATP for using the mitochondria is to break down glucose, which happens in the cytoplasm, not in the mitochondrion, and itself makes a little bit of ATP. But after that first step of glycolysis, you can then use what comes from a glycolysis in the mitochondrion to make lots more ATP, and most cells are going to do that. What do those cells that are making ATP and mitochondria also need to function? Yeah. What do mitochondria need to be able to make ATP from ultimately glucose? Oxygen, right. Erythrocytes, red blood cells, don't have mitochondria. They do not use oxygen to make ATP. They don't make a lot of ATP because they have to do it without oxygen, but they don't need a lot of ATP. They don't do a whole lot. They need a little bit. While this is actually mostly a joke, I think it's a good way of explaining it. Red blood cells do not have mitochondria for the same reason that Ben & Jerry's only employs lactose intolerant drivers. Yeah, if you're lactose intolerant, you can't eat the ice cream. Red blood cells can't use oxygen, so they carry the oxygen, but they don't use it. Now, in fact, that's not what Ben & Jerry's does. Ben & Jerry's has this more insidious plan to keep people from eating all the ice cream. They give them free ice cream. If you work for Ben & Jerry's, you get a free pint of ice cream every day, which I think the point there is that they get sick of ice cream pretty quickly, and so they don't want to eat it. But if you want lots of ice cream in your life, please go apply to Ben & Jerry's. You don't need to continue taking A&P2 if you're going to go to Ben & Jerry's. But no. So the idea of lactose intolerant drivers is a joke on my part, but it does illustrate the idea. Red blood cells can't use what they transport because they don't have mitochondria. And so it's the most efficient way of them to get the oxygen to their sources. Anyways, I got talking a little bit more about red blood cells, but we are in this flow chart. So all of these cells are made from this type of stem cell, except for the lymphocytes, which come from the lymphoid stem cell. And we'll talk about lymphocytes and the other white blood cells with the immune system in more detail. Hemopoietic, yeah, I guess that's how you pronounce it. Growth factors. This just means the hormones that are controlling the processes that were just there. Erythropoietin is the one that I mentioned when I was talking about testosterone a second ago. It's released from the kidneys. It's usually referred to by its abbreviation EPO. It stimulates the production of red blood cells. Thrombopoietin is another hormone that stimulates the production of platelets. And then cytokines are a class of hormones that are used mostly by the immune system, and so they're going to be important for making white blood cells. We'll talk a little bit more about cytokines as they function in the immune system when we get to that, but they are important for stimulating the production of those white blood cells. It goes into more detail about erythrocytes. I already kind of talked about them sort of in terms of the, Ben and Jerry's example, but here's a picture of hemoglobin. What it looks like isn't crucial to go through, but it does have 4 little bits inside, heme molecules, which are represented in this picture. And in the center of the heme molecule is an iron ion, which is what the oxygen actually binds to. So there are 4 subunits, sort of, there's actually 2 types, but there's 4 different very similar structures that come together to make hemoglobin. And each one carries oxygen in its heme molecule as iron, attached to iron. The life cycle of erythrocytes is important because probably one of the most toxic substances in your body is oxygen. Okay. Of course, you are probably well aware of the fact that if you don't get oxygen in your body, you will die. But oxygen, being a very reactive molecule or atom, can lead to biochemical errors and problems that accumulate in cells that are exposed to it. And red blood cells are exposed to the most oxygen, so they can have lots of big problems. And so they have a limited lifespan. A red blood cell lasts for about 120 days, about 4 months, in circulation, then it's recycled. New ones are made over and over again in the red bone marrow to replace the ones that have lived out their lifespan. This picture kind of describes what's happening in their life cycle. So they're born through the process of erythropoiesis, and reticulocytes, as they're getting rid of their nucleus, move into circulation and they become mature red blood cells, which circulate around for about 128 days. And then eventually they're going to be destroyed by a type of white blood cell called a macrophage, which ingests the red blood cell, breaks it down, and gets rid of its parts. The parts, some are recycled. So amino acids from the proteins and lipids from the cell membrane get recycled to make more of the same for cells in your body. The heme molecule gets removed and the iron separated out, which goes to the liver and red bone marrow to be incorporated in new hemoglobin molecules. The heme group, once the iron's been removed from it, gets modified into a different molecule, which is named here, and then it gets modified again and accumulates in the liver as something called bilirubin. Bilirubin is a component of a digestive juice called bile. So we get it from heme molecules from recycled red blood cells. But then bile goes into the digestive tract, and I'll explain what that does when we get to the digestive system. But as far as red blood cells concerned, it's made in the red bone marrow, circulates for about 4 months, and then is removed by a red blood cell and recycled or repurposed, and then the whole thing starts over again. There are several disorders of erythrocytes. The sort of best known one is sickle cell anemia. So here we have normal looking red blood cells, and here we have a sickle cell shaped one. Okay. So the sickle cell part of sickle cell anemia is describing the physical shape of the red blood cell. The cause of sickle cell anemia is a mutation in the hemoglobin gene, and one of the hemoglobin genes called beta globin, and the hemoglobin isn't properly assembled. It doesn't take on the shape that I quickly went past earlier, and it causes the cell to change shape. Okay. Because the red blood cell's messed up, it can't do its job, it can't carry oxygen, and that's what the anemia is. There are other types of anemia, but this is specifically the one that sickle cell is caused by a genetic disorder, and it causes cells to change shape like that. Now, if you get 2 copies of the sickle cell gene, one from mom and one from dad, you have the disorder. If you get 1 copy of the gene from one of your parents, but a normal copy of the gene from the other parent, you don't have the disease, but you're a carrier for it. And it turns out being a carrier or being heterozygous for sickle cell is a survival advantage against malaria. Because of having this 1 bad copy of the gene and what minor effects it has in a red blood cell, it does in fact make it harder for malaria to infect red blood cells. And it's protective that way. So this talks a little bit about the condition here and mentions a few others. But really the sickle cell anemia is sort of the #1 example of red blood cell disease that usually gets discussed the most. I'm going to talk about white blood cells with the immune system, and I'm going to save talking about platelets until I get to blood clotting. And so there's a section here on those 2 types of cells. The next section, called hemostasis, is not homeostasis, hemostasis. That's the formal name for blood clotting, and I'll talk about that in a little bit. What I want to do first, however, is talk about blood typing. And I'm going to use the book for a second to talk about it, and then I'm going to move to another website to show you some stuff about it. Now, the reason why this is important is it's a place where we're going to start learning about some things called antigens, which are going to be very important when we're talking about the immune system, and then antibodies that are related to that, and then blood typing is very important in considering what's happening in transfusion. Now, there's 2 aspects to blood typing. Okay? There's the ABO blood group and the RH blood group. When somebody says what their blood type is, they say that they're A or B or O or AB, positive or negative. Okay. The AB or O designation is the ABO blood group, and then the positive or negative designation is the RH thing. There are separate genes that handle this. I'm going to explain the ABO gene part real quick, and then I'll just say the RH is kind of like that. When we talk about genes and inheritance, there are different versions of a gene. So you have the gene for hair color, and hair color is much more complicated than I'm about to describe, but there are variations on that. There's gene variations that for blonde hair, for red hair, for brown hair, et cetera, et cetera. For blood typing, the ABO group has 3 different versions of the gene. There's the A allele, an allele is what we call a version of a gene, the B allele, and the O allele. What a gene is its instructions to make a protein. The A allele is instructions to make a protein called the A antigen. The B allele is instructions to make a molecule called the B antigen. And then the O allele is instructions that don't work, so no protein is made. Okay. Let me just write that down. The A allele is instructions to make the A antigen. The B allele is instructions to make the B antigen. And the O allele is a bad gene, and nothing is made from that. Okay. Now, usually when we think about simple genetics, there's two versions of the gene. Okay. There's a dominant version and a recessive version. And I got to get away from the hair color example a second ago because it's not simple. Here we have three versions of the gene. A and B are both dominant, and O is recessive. So it's what we call a codominant situation. And usually the dominant gene makes a protein and the recessive gene doesn't. Okay? If you get the A allele from one parent and the O allele from the other parent, then you have one functional copy of the A allele and you make the A antigen and you have type A blood. If you get the A allele from one parent and the A allele from the other parent, then obviously that's all you have and you make that antigen, you have type A blood. If you get the B allele from one parent and from the other parent, then you have type B. If you get B from one parent and O from the other parent, you still have one functional copy of the B allele, and so you make the B antigen and you have type B blood. If you get the O allele from both parents, then you do not have a functional copy of the gene and you have type O blood. Okay. One copy of the O allele does not give you type O blood. You have to have two copies. That's what's a recessive gene is about. Okay. Now, the codominant situation, the fact that there are two dominant genes here, means that there's another type of blood, which is if you get A from one parent and B from the other parent, then you have type AB blood. Okay. You have both genes. They make different antigens. Now, an antigen, the word means generating antibodies. And it doesn't generate antibodies in the sense of making them. Actually, when you're exposed to an antigen, your immune system makes more antibodies to deal with it. That's where antigen comes from. It's actually a kind of unfortunate term to use here because it's not exactly what it means. But the B antigen is something that is on the surface of the red blood cell. The A antigen is something that's on the surface of the red blood cell. And if your immune system is exposed to that antigen when you're not supposed to, then your immune system is going to react to it by making antibodies. So if you're type A blood and you get a transfusion of type B blood, getting red blood cells with the B antigen is going to cause your immune system to react to it, and you'll make antibodies and you'll reject the blood. Okay. If you get the A allele from one parent and the B allele from the other parent, then you make both antigens and you can receive A or B or AB blood as a transfusion, and your immune system won't react. And then O doesn't make any alleles, so there's nothing for the immune system to react to. So type O blood, anybody can get that because it's not going to interact with anybody's blood type.
Type O is supposed to be rare?
Actually, here's a breakdown of the numbers. So the bottom two are the positive and negative variants of O, and
So it's high in Latin American or
so it looks like it's actually one of the more common blood types. O positive makes up 47% of blood types for African Americans, 39 for Asian Americans, 37 for Caucasians, and 53 for Latino. O negative is very rare. So 4, 1, 8, and 4%. AB negative is even more rare than that. AB positive is kind of on equal footing. And you can kind of see there. Positive is much more common than negative across all 4 blood types. Okay. Now the positive negative thing, that's a separate gene, and there's only 2 of that. The allele for the positive blood type is dominant. The negative blood type is recessive, which would explain why positive is more common. You get 2 genes, one from mom, one from dad. As long as one of them is a positive blood type gene, you'll have positive blood. You have to get negative from both parents to have a negative blood type. And there's no codominance in that. Okay. If you put the 2 groups together, ABO and RH, then you get all 8 of these: A positive, A negative, B positive, B negative, et cetera, et cetera. Okay. Let's see. Here's a picture I wanted. So this table kind of shows you how all of this fits together. This is type A blood. It's covered in A antigens. Here's type B blood. It's covered in B antigens. And here's type AB blood that has both antigens on it. And here's type O blood that has neither of the antigens taken off. Okay. And this is just about the ABO stuff, not about positive and negative. Antibodies are a part of your immune system that reacts to antigens. If you have type A blood, you're going to have in your plasma antibodies against type B blood, or the anti-B antibody. Okay. If you have type B blood, you're going to have the antibody against type A. If you have type AB blood, you're going to have neither antibody. If you have type O blood, you're going to have both antibodies. Now, the reason behind this is a little bit involved, so let me explain it. When a human being is in utero, they get their immunity from their mother. And then after they're born, about 6 months to a couple years in, their immune system learns how to deal with its own environment. One of the stages of the immune system becoming mature is that it first learns what's supposed to be in the body. Okay? So it's tested for self-antigens. Before then, the cells that make antibodies, which are lymphocytes, make all the antibodies they possibly can. And everybody's going to make an antibody against the A antigen and the B antigen, just part of the human genome. But people with type A blood are supposed to have the A antigen. So in the first 6 months of life, the lymphocytes that make antibodies against the A antigen are destroyed so that you aren't allergic to your own blood, or you're not going to fight off your own blood. Same thing for type B. They're going to get rid of the lymphocytes that make the anti-B antibody. And so what's going to end up happening is somebody with type A blood will have the anti-B antibody. And somebody with type B blood will have the anti-A antibody. And for the other ones, people with type AB blood get rid of both types of antibodies because either one would react with their blood. Type O people get rid of neither antibodies because they don't have any antigens to worry about. Now, it seems kind of odd from the perspective of a modern human that this is the way the system works because it makes blood transfusions very tough. But this system works very well, for the natural biological systems, because what happens is your immune system makes antibodies against everything it possibly can. So you will potentially be immune to anything out there in your environment. But some of what it's going to make are antibodies against things that might be part of your body. And so in the first 6 months of life, your immune system learns what's supposed to be in your body and gets rid of the antibodies that are going to be autoimmune reactive. And so you just have left over whatever's left. If you don't have the B antigen, you're going to have the antibody against that. If you don't have the A antigen, you're going to have the antibody against that. If you don't have either antigen, you're going to have the antibodies against both of them. So the second row there, the antibodies in plasma, is just after somebody has a mature immune system, they're going to carry antibodies against things that aren't naturally part of their blood. And this system works great because without modern medical interventions, nobody's going to transfuse blood. People don't just naturally go up and bleed into each other. Okay. And in a situation where one person's blood does get into another person's blood in anything other than an intravenous delivery system, tube system, there's something wrong. And so the immune system wants to fight off the exposure to any kind of foreign blood. Okay. Nowadays, we understand that it can be helpful to transfuse blood, and so we want to do that. It's just we have to worry about how somebody's going to react to the blood that's transfused to them. So people with type A blood can get transfusions of type A or type O. People with type B blood can only get B or O. People with type AB blood, since they have neither antibody, can get any type of blood transfused in. People with type O, since they have both antibodies, can only get transfusions from other type O blood. And this doesn't even deal with the positive negative thing. Yes?
Is the AB including the negative AB also?
No, this is just the ABO. This isn't about negative positive. So if we add the negative positive thing in, somebody who has a positive blood type, they're going to have another antigen on their red blood cell, which is not depicted in this picture. And so they will get rid of the antibody against that other antigen called the Rh factor. If they have a negative blood type, which is technically what these pictures represent because there's no other antigens on them, they are going to have an antibody against the Rh factor. Okay. So people with a negative blood type have to get transfusions of other negative blood types. So down at the bottom, A positive people can get any A or any O transfusion. A negative people can only get transfusions of A negative or O negative. B, same thing. B negative can only get B negative and O negative transfusions. AB is the same thing. AB can get any of the ABO blood types, but if it's a AB negative, then they have to get a negative blood type transfusion. And O, same way. If you have a positive blood type, you will not have an antibody against the Rh factor. So you can get positive or negative blood type transfusions.
Negative is negative, but positive.
Can it be either?
So,
So it could be, if you have A net positive, you could get A negative or positive.
Right.
transfusion.
somebody with a positive blood type is not going to have antibodies against the Rh factor, so they can get positive transfusions also. But a negative blood type transfusion won't have the Rh factor on the red blood cell, so it can't react with anybody. So, yeah. Now, you can test for blood type by exposing a drop of blood to antibodies for those antigens. So if you're going to test for ABO and the Rh factor, you need 3 drops of blood. Okay, 1 drop gets exposed to the antibody against A, 1 gets exposed to the antibody against B, and 1 gets exposed to the antibody against the Rh factor, which until this book, I've never seen it referred to as anti-D, but whatever. It's the antibody against the Rh factor. Okay. The A and D drops here show reactions. What's happening is red blood cells are being clumped together by antibodies, and that's what you see in the drop. The B drop, there's no clumping. It's not reacting with the antibodies. Okay, so what this tells us is that this blood, which we have 3 drops of, has antigens, the A antigens on it and the Rh antigens on it. So this is A positive blood. This is just for testing. This is not about transfusions. It just lets you know what transfusions are possible. So this person who, when testing for blood typing, has anti-A reaction and anti-D reaction, means that they're A positive, they can get A positive, A negative, O positive, O negative blood transfused. They will have a bad time if they're given B anything or AB anything. Because, yeah. Now, this is done in the laboratory. What's happening is in the blood typing laboratory, they have 3 vials. So to that person's blood, they add a drop of the anti-A, a drop of the anti-B. Well, that's actually the anti-RH. They add a drop to each thing. This is not about what antibodies are in the person's blood, but what antibodies they're testing the blood with. So this person, in their blood, would have the B antibody. So they can't get any transfusions that have B in it. It's not what we see in the reaction, because the reaction is about what we're adding to the blood. But in this person's plasma, they're going to have antibodies that will reject a transfusion of anything that has B in the blood type. So, now, to go a little bit further with blood typing, in this folder, there's a blood lab quiz, which is going to be about the hematocrit and blood typing. I have a link here to a blood typing game. Okay, and I'll just turn the sound off a little bit. So it's loading the game, and it's showing this little thing. So it's ambulance being rushed to a hospital from some big accident or something, and there. They're wheeling 3 patients into the hospital. Okay. And then your job playing this game is to take each of these patients into the operating room or emergency department, whatever you want to say, test their blood and give them blood transfusions that they require. Okay. Now, there are 3 vials here. For the game, the way that this is designed, the antibodies look like these little squiggly things here. Antibodies have 2 binding sites to them. So one antibody can bind to 2 antigens, which the way they've drawn it here sort of highlights that it would be 2 separate cells that can bind to. But by binding to multiple cells, it causes the cells to clump together. So for the sake of these cartoons, this is what the anti-A antibody looks like, and it's in the vial that's labeled A. This is what the anti-B antibody looks like, which is in the vial labeled B. And this is what the anti-RH antibody looks like, which is in the vial labeled RH. They are drawn so that they fit like puzzle pieces with the antigens. And we'll see the antigens. Well, actually, I can go down here. So here's, well, this is A positive blood, okay? On the red blood cell, there are these blue scalloped looking things, which will react with the blue scalloped looking antibodies. For B blood, there's yellow spiky things, which will react with the yellow spiky antibody. And then in the O blood, there's nothing on the antibody. Okay, this is O negative blood. The positive antigen, the RH antigen here, looks like a little white square in the cartoon, which is going to react with this kind of H-shaped thing, which, okay, so this is the shape of that antibody. So 1 square can fit in there, and 1 square can fit in there. That's how it's depicted in the cartoon. So we can test her blood. To do that, you take the syringe, you put it in her arm, and you draw out some blood. And then you're going to put a little blood in the A vial, a little blood in the B vial, a little blood in the RH vial. Okay. Now you can look at the vials and tell what reactions happen. You can see a clump in the A and a clump in the B and nothing in the RH. But now also we can go and mouse over these vials and see the reactions. So here's her red blood cell reacting with the A antibody. So the blue scallopy antigen and the blue scallopy antibody fit together like puzzle pieces, and it binds two cells together, so we get a clump of cells when we do that. Same thing happens in the next vial. The yellow spiky antibody and the yellow spiky antigen fit together like puzzle pieces, and the cells clump together. However, in the other one, she doesn't have the RH antibody. She doesn't have the RH antigen, so there's no reaction with the antibody there. No clumping of those cells. So what type of blood does she have? Yeah. So she has a reaction in the A and she has a reaction in the B, so she has type AB blood. What else? Is it positive or negative? Negative. There's no reaction with the RH, which is negative. Now, really, the positive negative just means there's a positive reaction or a negative reaction. So no reaction means negative blood type. If there were a reaction, it'd be a positive blood type. Okay. So she has AB negative blood. We can take the AB negative bag and we can look at the cartoon there, and it looks just like the red blood cells we saw in the vials. Hang that here, and she gets a transfusion. You'll notice she has a kind of ashy face that gets better after she gets a transfusion, but you're being told that she needs more blood. Okay. Now we've used up the AB negative blood, so we have to give her something else. What else can we give her? Oh, what? Okay. So somebody said O positive. Why is that a bad idea? She has a negative blood type. She can't take a positive transfusion. So if we take the O negative, that's what she needs. Now in real life, if you had done that, accidentally given her a positive transfusion instead of a negative, she would have done more than vibrate, and you wouldn't have brought her back by giving her the proper transfusion. Okay. But for the sake of the game, when you make a mistake, just give her the correct thing.
Really, if they did give her the wrong type,
Right, because when in her blood, she's going to have clumping of blood cells just like in the vial there. And clumped blood cells are basically like clots. And she'll have a stroke or a heart attack or something like that. But they're saying she needs more. What else can we give her? We've used up the O and the AB negative. A what? She's negative, so we have to give her negative. We have to give her a negative. If we give her a positive, she's going to vibrate again. Okay. So that worked. But we still need to give her more. So what else can we give her? B negative. There we go. Okay. So now we've given her all that she needs. Conveniently, she needed 4 pints of blood, and there are 4 different types of blood that we could give her. So we exhausted all of our possibilities. I do this patient for demonstration in this game for everybody over and over again. I don't actually know or remember what the other patients are like. So you might not need to give them 4 bags, et cetera, but that's it. After you've done that, you can go back here and you have 2 more patients to grab. Do the same thing. Test their blood, figure out what they need, give them the correct blood type. Okay. I'm not going to finish off this game. You'll be doing that yourself. There is a back button here, which you are not required to go to, but I'm going to show you where this is coming from. You will notice in the top corner here, it says nobelprize.org. This is presented by the Nobel Prize website. If we go back here, it takes us to the main article for blood typing, or actually the blood typing game. What I've done, the link I've given you in Blackboard, which is the one you need to use, takes you to the old blood typing game. Okay. If for some reason you want to do it from the Nobel Prize website itself, you have to go to the bottom of this page and click on the link to the old thing. And then it's an old page and they haven't kept it up, so it doesn't quite work. But move the mouse around down here and you'll find there's a link here. Well, first off, you want to go to the game. Oh, that's right. It's an old page, so there's lots of problems. And for some reason, it can't read Flash. But we just played the game. We know that I have Flash on this computer, but the page doesn't work. But there is a link down here to an article that explains blood typing in some detail, which I've often used to explain all this, but the book does it pretty good, so I just went through there. This is just a picture describing the blood's made up of plasma and cells, and they've broken down to red, white, and platelets. And then here's the different ABO blood types, a little explanation of that. Weird quirk here, if I use the Chrome browser to show you this page, little cartoons that show the blood types are too small to see, but it's the same cartoons I just showed you in the game. And then they explain the RH factor, and the cartoon pictures here are big enough to see, but whatever. A little something to say about the RH blood types. And then here are the 8 types that we get by putting A, B, O, and positive, negative together. You also can't see a little animation here that shows what happens when you combine A positive and B positive blood. So just a suggestion of the kind of thing that would happen there. If you're interested in seeing this, you can open this up in Firefox, or I imagine Internet Explorer, and it should work, but for some reason in Chrome it doesn't. The clumping that I've been talking about is called agglutination. Okay, agglutinate is the verb form of that, and the book uses that term to describe it. And then it goes on talking about blood typing, transfusion. Here's a big table that shows all 8 blood types, what antigens each person has, what antibodies each person has, what blood they can give to, what blood types they can donate to, and what blood types they can receive. So AB positive can receive all 8 blood types because they have no antibodies. They're not going to react to anybody's stuff. O negative can give to all 8 blood types, okay, because they have no antigens on the red blood cells of the blood type. Now, while O negative can give to everybody, it can only receive other O negative blood. While AB can get from everybody, AB positive can get from everybody, it can only give to other AB positives. Okay, so there's a trade-off there. O negative is often thought of as the universal donor. Okay, if you have O negative blood, you are probably aware of that, and the Red Cross is too. Okay, so you probably get a call every few weeks saying, "Hey, you want some ice cream? Not ice cream. OJ and cookies. Come on down, hang out with us for a while," and they'll take your blood. O negative is a very rare blood type. It'd be great if we get a bunch of it, but there's not really enough people to donate. So O positive is useful if we can just establish if somebody is negative or positive. Positive blood types can get that as a universal donor too, since it's really the most common type. It's just people who are negative, it's an issue, but negative blood types across the board are kind of rare. Yeah.
So if there's a reaction in the sample for the different types of blood, let's say an A, that means that person has that type of blood.
Right. So when you're testing it, the reaction of clumping tells you that the blood cell has that antigen on it.
And that's what they are.
And yeah, so there's a reaction in A, then they're type A blood. If there's a reaction in B, then they're type B blood. If there's a reaction in both, then they're type AB blood. If there's a reaction in neither, then they're type O blood. And then with the third drop, if there's a reaction, it's a positive blood type. If there's no reaction, it's a negative blood type. Now, if you look closely at what this says, which I doubt you can do from where you're sitting, under the antibodies thing, it will say for the negative blood types can develop Rh antibodies. Somebody who is a negative blood type doesn't necessarily have antibodies against the Rh factor. But when they're exposed to positive blood type, they'll probably develop a sensitivity to it. So having a lot of O positive blood on hand can be helpful even for negative blood types. It's probably very dangerous to do, but if the choice is between somebody dying and seeing if they're sensitive to a positive blood type, it might be worth the trouble. I am not in the medical field by any stretch of imagination. I'm not advocating to do this, but given the numbers we're looking at, it is possible that O positive blood type can be useful to have as a universal donor on hand, just because of it. And as an example of how somebody with a negative blood type can tolerate positive blood types, at least their first exposure to it, you can look at pregnancy. Now, when a woman's carrying a child, the fetus is getting its nutrition through the mother's blood supply. So there's some interaction between the fetal and maternal blood. If the mother has a negative blood type, but the fetus has a positive blood type, then the mother's going to be exposed to the Rh factor. And this can happen because if the mother has a negative blood type, she's going to pass the negative blood type gene on. But if the father has a positive blood type, he's going to pass the positive blood type gene on, and the kid will have a positive. Because
What about with twins? Would the twins have the same blood type?
it depends on if they're identical or not. So, but it's still the same idea. It's just about inheritance. So the mother is negative, she can only pass on the negative gene. But if the father is positive, he might only pass on the positive gene, but if he carries the negative gene, then 50-50 chance whether he's going to pass it on. So it's possible for the mother to have a negative blood type and the fetus to have a positive blood type. And if that happens during the first gestation, it's probably not going to cause problems because the woman's probably not sensitive to the Rh factor. She doesn't have antibodies. But after exposure to it, she will develop antibodies. As a matter of course, when a woman with a negative blood type gives birth to a positive blood type child, she will get treated for that Rh factor sensitivity, so that if it happens again, if she and her husband decide to have another child and the same thing happens, she will not be sensitive to the positive blood type of the fetus. If she is sensitive to the positive blood type for the second gestation, it will not come to term. Her immune system will reject it. This is something that was probably only understood in the past, 40 or 50 years. Of course, humans have been having babies for a lot longer. And it just kind of blows my mind that there are probably people out there that were trying to have kids, and they might have had one, but could never have a second kid. And it just came down to this, that the father was passing on a positive blood type gene and the mother was negative, and after the first child, she developed a sensitivity to the blood, and so she rejected the fetus every time. Very simple intervention these days, it can be handled, but just kind of blows my mind that for so much of human history, that was probably the underlying cause for people not having more than one child in a marriage or something like that.
So is that where the Rogum shot?
Sorry?
Is that where that Rogum shot?
Right, I think that's what they're called. Anyways, so this is just a little bit more explanation of things if you want to go in to look at it. Use the link to the blood typing game that's here. If you don't, if you go to nobelprize.org and look up blood typing, it'll take you to this page, and if you play this version of the blood typing game, it won't work. Okay. Now it'll work. It's a great game. What they did, so the version I just showed you, those 3 patients always have the same blood type. The new version, they've set it up so it can do all 8 blood types, and it just randomly selects 3 of them out for each time you play. Now, the reason this is important is that in the blood lab quiz, you have a question on the results from the game. That question is dependent on you playing the version of the game that I linked directly to at Blackboard. If you play this version of the game, you will not be able to answer that question. Or the chances of it are like 1 in 350 times. So if you want to play the game 350 times and hope you get the question right, that's fine. But use the old version of the game because the question set up the old version. Also, the new version, the animation's a little kind of, I don't know, freaky. You can see that's what it looks like. So they took a photograph of somebody's head and a photograph of somebody's arm and photoshopped that in with drawings of, somebody lying on a table. And it just looked kind of weird. So at least the very stylized cartoon version of the game, sure, one of them has like a spiky mohawk or whatever, but at least it's not quite as disturbing as this version of the game. So, but don't play this one, play the new one, which I linked directly to out of Blackboard. And it's in that link is right before the Blood Lab quiz because you need to complete the game to answer all the questions in the quiz. Okay. The other thing I want to talk about with the blood is blood clotting, which is the section just before what we were just going through. But I want to be sure I covered that completely because it's the blood quiz needs that. Hemostasis is the formal name for blood clotting. Hemo for blood and stasis, like to stay. So it makes the blood stay in circulation. Don't confuse that word with homeostasis. They mean completely different things. Although you could say that hemostasis is a homeostatic mechanism, but that gets kind of confusing to deal with. There are three major parts of blood clotting. Okay, when blood starts coming out of the circulatory system, immediately what will happen is a vascular spasm. The muscular wall of the blood vessel that's damaged will contract. Okay, quickly, it's a spasm. And by narrowing the damaged blood vessel, it limits the amount of blood that goes through the blood vessel and therefore limits the amount of blood that you're going to lose. And then you start forming a plug to stop the bleeding that's made out of platelets. This is a temporary blood clot. Okay. After the platelet plugs formed the temporary blockage, then the actual blood clot, what we think of as a scab if it's on the surface of your skin, is going to form. And that process is called coagulation. Formation of platelet plug and coagulation are actually kind of taking place at the same time. It just platelet plug formation is very, very rapid, and it stops bleeding immediately. Coagulation takes a little bit of time. And so it starts immediately. It just has to take a little bit of time to get to the point where it's going to be able to hold back the loss of blood, but it's going to end up helping the tissue heal up wherever the damage is. Now, this picture, after where it says coagulation, puts this all together and explains the process. Okay. So it starts with some sort of damage to the blood vessel. Okay, so you cut yourself, you cut through a blood vessel, and blood's coming out of it. Okay, the vascular spasm is just when the muscular wall of the blood vessel contracts, which is going to narrow the blood vessel. So just the volume of blood passing through decreases, and therefore the volume of blood that you're going to lose is going to decrease also. And it can make for an easier site to form the blood clot, smaller space, and it can heal up or something like that. Okay. And then at the site of the damage, so this is the damaged end of that thing, it narrows down because of the vascular spasm. Platelets are going to start forming a plug where the blood's being lost from. Okay? Now, in this picture, we see two forms of platelets. This form over here is the normal form of a platelet, just floating around doing nothing. And then this arrow shows a regular platelet becoming an activated platelet. And activated platelets are what form the clot. Okay. And when platelets are activated, they start to release chemical signals to help activate other platelets. And they start to aggregate together and make the plug, okay, which is going to seal off where blood is lost. This process is one of the very few positive feedback cycles that you have in your body. Okay. The platelet is activated. And once it's activated, it releases a chemical that will activate other platelets. And when those platelets are activated, they release a chemical that will activate other platelets. And when those platelets are activated, they release a chemical that will activate other platelets. When those platelets are activated, they release a chemical that will activate more platelets. When those platelets are activated, they release a chemical that causes more platelets to be activated, et cetera, et cetera. Okay. The response increases with each iteration. Okay. Why is that important?
To release the chemical platelet?
So that's what's happening, but why is it important for that to happen to cause what?
To form the clot.
And how is that going to help form the clot? By having this positive feedback signal, where each activated platelet activates more platelets.
Does the muscle contract?
No, that's just about the platelets. Now, when I was a kid, which nowadays probably means none of you were around for this, there was this commercial. I don't remember what shampoo it was, but it's a shampoo commercial. And it started with a picture of a woman's face, and she said, "Oh, I just tried this new whatever." She doesn't quite do it as silly as I'm saying, but and she says, "I told 2 friends," and then her face on the screen splits into 2 copies of her face on the screen. She says, "And I told 2 friends," and they split into 4. And then those 4 say, "I told 2 friends," and it splits into 8, and those 8 spread into 16, et cetera, et cetera. That's the idea. When one platelet reacts, is activated, it releases chemicals that's going to activate more platelets. Okay, and when those platelets are activated, it's going to release chemicals that activate more platelets. When those platelets are activated, you're going to get more platelets activated. Okay. So it's going to cause probably an exponential increase in activated platelets, which are going to then congregate at the site of blood loss and form this platelet plug. Why is that important?
You just stop bleeding.
Right. And it makes it, you stop bleeding very quickly. Okay. So if you activated a platelet and it went off to do its thing, but it didn't activate more platelets with it, then it's going to be a very slow process to get all those platelets activated. Positive feedback creates very, very rapid responses, okay, which is going to limit the amount of blood that's lost at the site. Okay. Now, if it was just about the platelets, eventually you'd have too many platelets and it'd cause a big problem. When blood loss ends, there are going to be signals that shut down the clotting process. So it will end eventually, but that's what gets it all started. And it's a positive feedback. Then coagulation is when a protein mesh forms around the site of blood loss and forms more permanent clot. Now, the mesh that's drawn into the picture there is around activated platelets. So the platelets that have been activated and been causing this platelet plug get incorporated into the more stable clot, or the fibrin mesh. And in the scanning electron micrograph here, this is a platelet, but really you also see a lot of red blood cells. In this picture, they might have, what, a couple of red blood cells there, but there's so many red blood cells, they get caught in the clot too. And that's why clots usually look red. Okay, so if you have a scab on your skin, it's red because it's got red blood cells trapped in it. Okay. They don't have anything to do with the actual clotting. There's so many of them, they're going to get caught in the clot. But then the fibrous strands that you see in here, these are protein strands that are locking together and making this mesh that is the coagulant. Now, for the picture here I just zoomed in on, it shows the process with drawings. We have vascular spasm, platelet plug formation, and coagulation. Over on the other side, not perfectly lined up with the events that are happening over here, but the idea is that this is sort of the time course. You have the series of clotting factor proteins that are involved in this. Okay, so each of these little squares or ovals represents a protein which is called a clotting factor. And these are proteins that are in your blood that are always there. And when something happens, one gets activated and then it activates the next one. It just leads to a cascade of events. Okay. Now, the clotting factors are numbered usually by Roman numerals. So we have like clotting factor 1 through, I think it's 13 or something like that. I don't care that you know the numbers that are in here. Just, I like the depiction here. One thing becomes activated and it causes the next thing to be activated, which causes the next thing to be activated, or whatever like that. Okay. Now, what's depicted here is there are actually two different pathways that lead to clotting. One's called the intrinsic pathway and the other's called the extrinsic pathway. Now, the book actually kind of gets this a little confused. So let me explain a little bit better. The intrinsic pathway is describing things that are happening in the blood. That's what intrinsic is referring to, within the blood. So it's really about the platelets and the clotting factors that are in the blood. Extrinsic pathway is referring to things outside of the blood. And this is where the book kind of gets things screwed up a little bit. Under extrinsic pathway, it says trauma to extravascular cells. What that really should say, if it's even a real word, would be trauma to extra hemo something cells. So it should be damage to cells that are not part of the blood. What that's saying is damage to cells that are not part of the blood vessel, but it's actually the blood vessel cells that are the root of the extrinsic pathway. The intrinsic pathway is platelets getting activated and activating themselves and that sort of thing. The extrinsic pathway is when there's damage to the blood vessel, how that initiates blood clotting. And essentially what that is that the connective tissues in the wall of the blood vessel, which contain collagen, when they're exposed to platelets, they're going to activate the platelets. Collagen is found in every connective tissue except for blood. And there's no collagen in blood. So when when blood is exposed to collagen, that's wrong, and the blood reacts to that by clotting. That's really what's going on in the extrinsic pathway, although the book kind of says it wrong. For the intrinsic pathway, platelets can get activated by any sort of trauma that happens to them. If two platelets run into each other, they'll activate each other. That's intrinsic pathway. If there's damage to, if there's trauma to a blood vessel that doesn't actually cause damage, that can potentially start activating platelets and start the intrinsic pathway. Or if anything happens that two platelets run into each other, and if you've ever seen a Plavix commercial, you've actually kind of seen this. This is the whole point behind the drug Plavix. If somebody gets a cholesterol plaque in a blood vessel and it narrows a blood vessel, the platelets will start running into each other, and that can cause the intrinsic pathway to be activated. So let's say this is a blood vessel, okay, and blood's supposed to be, moving through normally like this. If you start to get a buildup of cholesterol on the inside of that blood vessel, then the blood's going to have to start squeezing through a more narrow area, and platelets are going to start running into each other, and you're going to get the initiation of blood clots there. Plavix just keeps that from happening. You don't want a clot forming in that situation because there's no loss of blood. It's just a quirk in the system that's going to start activating platelets and initiate blood clotting. The intrinsic pathway is actually kind of slow if it's working on its own, because sometimes it can be activated by accident, and so there's going to be enough time for it to sort of shut itself off. Did I say that right? The intrinsic pathway takes a long time, can be shut off. Right? The extrinsic pathway happens very quickly because when the extrinsic pathway is activated, you are losing blood. Okay? That's damage to the cardiovascular pathway, circulation. So let's go back. I said, let's say there's trauma to a blood vessel, but not damage. Okay, so somebody's blood vessel is getting squeezed. That's going to initiate the intrinsic pathway. That trauma continues to the point where the blood vessel is actually damaged. The intrinsic pathway has already started being activated, and the extrinsic pathways are going to jump in, and you get blood clotting forming from that. So the intrinsic pathway is there to sort of start getting things ready if trauma's beginning, but the extrinsic pathway is really the clue that there's some serious trauma involved. Whether it's the extrinsic pathway or the intrinsic pathway, they both eventually converge on what's called the common pathway or the final common pathway. This is the actual formation of the blood clot. So everything leading up to this is just a series of reactions that get it ready. But then the final common pathway is actually making the blood clot. There is one of the factors there, and I can't remember offhand which number it is, and I can't read it off the screen from where I'm standing, but one of the factors that's numbered, it's probably 10. So factor X, which is actually Roman numeral 10. It ends up causing a protein called prothrombinase to become thrombinase. No, I said that wrong. Prothrombin to become thrombin. When you have pro in front of something, that means that it's a precursor. So prothrombin is a precursor to thrombin. And what clotting factor 10 does is it stimulates that conversion of prothrombin into thrombin. Thrombin is an enzyme. And what thrombin does is it converts fibrinogen, which is one of the major plasma proteins, into fibrin. Fibrinogen is a soluble plasma protein. It's dissolved in the plasma. Fibrin is an insoluble protein. So when thrombin converts fibrinogen into fibrin, it makes it into an insoluble protein, which means it no longer dissolves in the plasma, and it falls out of solution. And it does that at the site where the clot needs to form, and so it's available to become part of the clot. And so at the end of this arrow here, it says cross-linked fibrin clot. Fibrin threads, fibers, end up being worked into this mesh that becomes the clot itself. So again, I don't really care that you know the specifics here, just the idea that there's an intrinsic and an extrinsic pathway that both converge on the final common pathway. And then for the final common pathway, I do want you to know this. I think it's factor 10 or maybe it's factor 5 that specifically causes the conversion of prothrombin into thrombin. Prothrombin's the inactive form, thrombin's the active form. And what thrombin does is it causes fibrinogen to become fibrin, which then forms the clot itself. Don't worry about this factor 13, but it's part of the fibrin clot formation. I'm just interested in knowing these things. Now, something that's not in this picture is the fact that thrombin has another role to play, which is another site of positive feedback. And to show you that, I actually want to go to the presentation I made for this stuff a while back, where I have pictures from another book, but saying the exact same stuff. And then I have some text slides I want to show you. Come on. Okay, so if you go to this presentation, which is in the Prezi link in the syllabus, if you're interested in it, you're welcome to look through the whole thing. All of the other stuff about the blood system is in here. I'm just interested in talking about hemostasis here. And it says vascular spasm, platelet plug, coagulation, just like I said. And here's pictures from another book of the same sort of stuff. So here's the vascular spasm where the blood vessel gets narrow after it's damaged. Here's platelets forming a plug, and these are activated platelets. The current book's picture is a little bit better just depicting that. And then here's this book's version of what I was just showing you with the extrinsic pathway and the intrinsic pathway converging on the common pathway. So here's that factor 10 I pointed out. I think what in the current book says factor 5 here is called prothrombinase, which is a word I said a second ago by accident. When it ends in an ase, that means it's an enzyme. And what prothrombinase does is it converts prothrombin into thrombin. Okay? And then thrombin converts fibrinogen into fibrin, which becomes the clot. Thrombin also will feed back, and when there's an increase in thrombin in the system, it's going to activate more prothrombinase, or it's going to activate more, I think it's platelets in the intrinsic pathway, which is going to increase what's going on here. So prothrombin is converted into thrombin, which can make the clot, but it also goes back and activates prothrombinase, which converts more prothrombin into thrombin. And therefore, the thrombin is available to make more prothrombinase to convert prothrombin into thrombin, which makes thrombin able to make more prothrombinase to convert prothrombin into thrombin, which makes more prothrombinase, another positive feedback step. So just like platelet plug formation, which is a positive feedback step, thrombin is also capable of positive feedback, which is going to speed up coagulation time. Again, to limit the amount of blood loss and get the tissue repaired quicker. In this picture from the other book, it also doesn't actually show that. So I have some text slides here that explain all this. First off, a quick recap of what the extrinsic and intrinsic pathways are about. The whole of the common pathway, prothrombinase is formed, which was not in our book. It's, I think, what the book calls factor 5. Prothrombin is converted to thrombin. Thrombin converts fibrinogen to loose fibrin threads, which are insoluble, and stabilizes them into a clot. Thrombin also is involved in positive feedback cycles. It activates more prothrombinase. It also increases the intrinsic pathway activity, which is going to cause more prothrombinase to be active. There's actually two things that thrombin does, both having the same effect, but it can be active in two different ways. And then, the making that fibrin clot then makes it possible for the edges of the damaged tissue to be pulled together, and it can heal up a little bit better. Epithelial cells making the line of the blood vessel or in your skin or wherever it might be, they heal very well. Epithelial tissue is just very good at healing. Connective tissues don't heal as well, or maybe they heal, I should just say they heal differently, in that they form scar tissue. Basically, extra fibroblasts form in that area, lay down extra collagen in the ground substance, which is the clot tissue. If it's a small site of damage, there won't be a big clot, but if you cut through your skin severely, then you'll form a pretty big scar there, which is really in the connective tissue part of the skin. Okay. Well, the clot makes it possible to pull the cut tissue together, the damaged tissue together, allowing it to heal up there. So again, I just want to point out to you this presentation I have on blood that includes up here in this corner a little bit more detail on the blood clotting process. It's not any, well, it's a little bit different from what the where'd it go? What the book shows. So all of this is in the stuff I just showed you, but I added in the text slides that I point out there that thrombin will feed back and increase the production of more thrombin. And then that more thrombin can feed back and increase the production of thrombin. I can do that directly here, or it actually goes back into the, well, I should say intrinsic pathway to do that sort of stuff. And just those positive feedback signals, two of them within what thrombin does itself, and then additionally, the platelet plug formation is its own positive feedback cycle. They just speed up the formation of clot and therefore limit the amount of blood lost and how quickly the tissue can heal back because of that. So that's the stuff about blood that I wanted to cover a little bit more. Any questions on that?
Are those the only two positive feedback that happen in the blood system? Is that
Right. So it's, I mean, where it's happening is important. It's happening in the two places where we actually stop blood loss. Either the temporary platelet plug has its own positive feedback, or in the final common pathway where we make the more stable clot, it has its own positive feedback. And thrombin actually has two targets, both of which are going to cause an increase in thrombin activity. So. So we've got almost a half hour left today. I want to at least start talking about the heart. And we'll, of course, spend more time tomorrow talking about the heart, but I want to introduce it to you here. Let's just go here. What I want to start with is to just talk about heart anatomy a little bit. First off, the heart, if you weren't aware, is in the thoracic cavity. Specifically, the region where it's found between the two lungs is called the mediastinum. It's oriented slightly to the left. It kind of comes to a point over here on the left side, which is called the apex of the heart. The apex of the heart is actually the lowest point of the heart, but if you sort of think of it as coming to a point there, that's sort of what apex is getting at. The opposite side of the heart, which is where it's connected to everything else, is called the base of the heart. Base and apex usually go together. So we have that there. There is, in fact, a gene that tells the heart to develop a little bit onto the left side, having the very inventive name of lefty. I know of this for two reasons. One, when I was in graduate school, it was discovered. And so it's kind of interesting news about development, because I was a developmental neurologist at the time, that there's a gene that specifically says this is the left side of the body, so things on the left side should develop that way. The other reason why I'm familiar with it is that there's an episode of CSI where somebody gets shot through the chest and it doesn't kill them, and so they have to figure out why that is. And it turns out what it is that the person has their heart on the right side, which is in fact an actual thing. If somebody has a mutation in the lefty gene, their heart will develop on the right side. It's very, very rare, but it makes for good television apparently. So, but yeah, for the most part, it's found mostly on the left side. Okay, it's a midline structure, but it points off to the left side. Let's see. Let me enlarge this picture here. Can't quite get it there. I just want to zoom in on this, but I want to try to get it all on the screen. So the important thing to get out of the anatomy of the heart is that there are 4 chambers to the heart, 4 valves in the heart, and 4 major vessels attached to the heart. The chambers, there's 2 on the right and 2 on the left, and 2 that are at the base and 2 that are towards the apex. The ones that on the base side of the heart are the atria, top of the heart, and the ones towards the apex, bottom of the heart, are the ventricles. So you have a right atrium, a right ventricle, a left atrium, and a left ventricle, 4 chambers. Atria, which is the plural of atrium, receive blood coming from somewhere else in the body. And then it goes from the atrium into the ventricle, and then the ventricle pumps that blood to the next target in the body. The right side is responsible for receiving used blood from the body and pumping it back to the lungs to be reoxygenated. The left side receives that oxygenated blood from the lungs and pumps it back out to the body. In this picture, the right side is depicted in blue and the left side is depicted in red, which has to do with the oxygenation state of the blood. So the used blood from the body, it's not actually blue, but that's the way we usually think of it, because it has a low oxygen content. It comes into the right atrium through vessels called the vena cava. There's the superior vena cava and the inferior vena cava, which is just a matter of upper and lower body. So the superior vena cava is bringing blood from the head, neck, shoulders, and arms, everything above the heart. And the inferior vena cava is bringing everything up from the legs and the abdomen and the lower trunk. So both of those feed into the right atrium, and then the right atrium goes into the right ventricle, and then the right ventricle pumps the blood up towards the heart into what's called the pulmonary trunk of the pulmonary arteries. One pulmonary artery goes to the left side, one goes to the right side, and going off to either side then branches into more stuff. But it's carrying that deoxygenated blood to the lungs. And then in the lungs, it'll swap out carbon dioxide for oxygen, and the newly oxygenated blood will come back to the left side of the heart through the pulmonary veins. So there are 2 pulmonary veins on the left and 2 pulmonary veins on the right. They never come down into one from either side. There's 2 from either side, and they come into the left atrium, and then and you see these 4 white lines in the left atrium here. That's supposed to depict coming from 2 right pulmonary veins and 2 left pulmonary veins. That all goes down into the left ventricle, which then pumps the blood up into the aorta to go out to the body. Okay. The heart really is 2 pumps. The right side and the left side are separate, ideally. The right side is pumping deoxygenated blood from the body to the lungs. The right side is pumping oxygenated blood from the lungs back to the body. There's a wall separating the 2 sides from each other called the septum. When we're talking about the wall between the atria, we call it the atrial septum. When we're talking about the wall between the 2 ventricles, we call it the ventricular septum. I said ideally that they should be separate. There are some conditions where there are holes between the 2 sides in the septa, and so there can be a mixing of the blood from one side to the other. But ideally, they should be separate. To make sure the blood's flowing properly through all of these structures, we have valves. The point of a valve is to ensure that blood flows in only one direction. Valves are kind of like funnels. Okay, so they have a wide open end and they have a narrow end. I'm exaggerating a bit, but sort of like this. So if blood is going to go from an atrium to a ventricle, the atrium side of the valve is going to be wide open and the ventricle side is going to be pretty narrow, and the blood's going to go down. Now, once the blood gets into the ventricle and it fills up the ventricle, it's going to push back against the valve and cause it to close. Okay? So if we look at this picture here, you can kind of see the funnel shape suggested by the valve. As the blood fills up the right ventricle here, it's going to push back against the valve, and it's going to push the 2 or 3 leaflets together, and it'll close up. That's going to make sure that the blood doesn't go back into the atrium here. It's going to ensure it only goes one direction. Now, there are 4 valves, and let me write this part down because it gets a little complex. There are 4 valves, and we describe them as 2 different groups. we have atrioventricular valves, and we abbreviate atrioventricular AV, okay? And I'm putting the AV abbreviation after the word atrioventricular because that's what it's abbreviating. It's not atrioventricular valve, okay? It's AV for atrioventricular valves. So because atrioventricular can be a little bit of a mouthful, I'll mostly refer to them as AV valves. Okay. There are two of them, one on the right and one on the left, which I'll get to in a second. And then we have semilunar valves, which for the sake of being consistent would be abbreviated SL, but that's usually not used too much because it's just as easy to say semilunar. So on the right side, the atrioventricular valve there is called the tricuspid. And on the left side, it's the mitral valve, which is also sometimes referred to as the bicuspid valve. And then the semilunar valves on the right side, we have the pulmonary valve. Actually, valve on these other ones. And on the left side, we have the aortic valve. Okay. So, atrioventricular valves, as you can tell by their name, are between an atrium and a ventricle. Okay. So on the right side, between the right atrium and the right ventricle, we have the tricuspid valve. And on the left side, between the left atrium and the left ventricle, we have the mitral valve, which can also be referred to as bicuspid. Semilunar valves, the name comes from the fact that they look kind of like a half moon or semi-moon. Okay. On the right side, or sorry, before I get there, they are found between a ventricle and an artery. On the right side, we have the pulmonary valve, and on the left side, we have the aortic valve. They're named for the artery that they're adjacent to. Okay, so on the right side, from the right ventricle, blood goes through the pulmonary valve to the pulmonary trunk. On the left side, blood goes from the left ventricle through the aortic valve into the aorta. It would look like from these names that tricuspid is a special name for that valve. There's something special about it having three cusps. It's in fact not true. All valves have three cusps except for the mitral valve, which has two, which is why it's also known as the bicuspid valve. I have no idea why, but for some reason, just historically, tricuspid became the name for that valve. Although the pulmonary, aortic, and other valves all have three cusps to them also. That one just got named tricuspid valve. Now, the mitral valve is called the mitral valve because it looks like a mitre hat, or a mitre hat. A mitre hat is most commonly known as the Pope's hat. Okay? So have you ever seen the Pope? Well, the current Pope doesn't wear it anymore. But back when popes liked pomp and circumstance, they'd wear this hat that came up to kind of a point. Okay? And it was really two pieces of cardboard, whatever Pope hats are made out of. One piece in the front and one piece in the back. Okay? It has two sides. And so the bicuspid valve is the same sort of thing. Okay? It has two leaflets. So it's kind of like a mitral, it's kind of like a mitre hat. That's where the name comes from. It would seem more reasonable to just call it the bicuspid valve, but more often it's referred to as the mitral valve, especially in clinical situations. So I'm going to refer to it as mitral because that's just the name that you're going to hear most often. But understand that in naming things, there's often multiple names for things. Sometimes there's an anatomical or a physiological name for things. Sometimes one person comes up with a name, the next person says, but I want a name, and comes up with their own name, and everybody picks a second name. So there's sometimes multiple names around. Deal with it. So you might see bicuspid, but more often you'll see mitral, and I'll try to stick to mitral here. Okay, so those are the valves. Going back to the picture here. So here's the tricuspid valve between the right atrium and the right ventricle, and then here's the pulmonary valve from the right ventricle into the pulmonary trunk, which then branches right and left to the pulmonary arteries. Pulmonary trunk is an artery. It's just the singular base of the pulmonary arteries. And then on the left side, from the left atrium to the left ventricle, we have the mitral valve. And then from the left ventricle, blood goes to the aortic valve to go up into the aorta. Now, in this drawing, things are a little bit weird because it's a two-dimensional depiction of three-dimensional heart. The pulmonary trunk lays, I should say this way. Nope, I should say this way. So pulmonary trunk going from the right side up lays on top of the aorta, which is going left side up. Okay. Actually, the aortic valve would not be visible from this point of view because it would be just deep to the pulmonary valve. They're basically right on top of each other. But for the sake of the picture, they kind of squeeze it in. So right side pumps this way, left side pumps this way, and those two vessels just cross over. Okay. You can follow blood flow through the whole system, and I'd like you to get familiar with this. What I would normally do is, after explaining all this for lab, I get the heart models out, and you would look at those and find all 12 of these things, the four chambers, the four valves, and the four vessels, on the three-dimensional heart model so that you can orient yourself to that. I'm not going to do that today because we're near done for the day. What I'll do is at the beginning of next week's class, I'll have the heart models out. Before we get started, go ahead and grab a heart model and look at it. Okay, so those of you that are in early, you'll have plenty of time to look at the heart models and get to know it. Okay. To finish out this picture, I just want to, talk about the rest of what we see here. So here's the heart drawn as anatomically correctly as possible here in the middle of the body. And we still have the blue and the red representing deoxygenated and oxygenated blood. As the heart's pumping blood out to the body or to the lungs, in those structures, the blood vessels will come together as capillaries where oxygen and carbon dioxide will be exchanged. And it sets up what we think of as two different circulation circuits, if you will. In actuality, it's all sort of one circuit. It's more like a figure eight. But we have the systemic circuit, which is the freshly oxygenated blood being pumped from the heart to the body. And then after it's been deoxygenated, it comes back to the heart. And then we have the pulmonary circuit, which is the deoxygenated blood being pumped from the heart to the lungs, and then when it's reoxygenated, it comes back to the heart. Okay. So I think the pulmonary circuit is pretty obvious here because we can see sort of the outline of lungs on the other side of the heart. The systemic circuit is represented by the two loops, one going up and one going down, and they're just representing the systemic circuit going through the lower body and the systemic circuit going through the upper body, but without showing all of the head and neck and all that. This model on the wall over here kind of shows the same thing. And you can, the way it's depicted in the picture on the screen there, it's more obvious where the loops are. But here's the heart in the middle. The pulmonary circuit is just going to be blood going from the heart to the lungs and back to the heart. Or I should say, from the heart to the lungs and back to the heart. The systemic circuit is going to go from the heart out to the body, whether it's inferior or superior, and then back to the heart. And this shows a little bit more of the vessels around. When we talk about the vessels next week, about time, we might talk a little bit more about this model. But this kind of shows what these things are. Now, I would like to leave you with a little video. Okay, for those of you that got the reference about the shampoo commercial, I told two friends and they told two friends, you probably recognize that too. That was from Happy Days, and which was a sitcom a long time ago, where Henry Winkler was best known as the Fonz, and Ron Howard played Ron. Oh, what's his name? Richie Cummings. It was originally when they were in high school, but of course the show went on for a long time, so they ended up going to college. That's them in college, and they're in their A&P class. Potsy, who was the main singer there, was having trouble with learning the cardiovascular circulation in the class, and so he went to the Fonz, which I'm sure is everybody's choice to get academic support from. But the Fonz made a very good suggestion, which is to use what he knew to understand the material. And Potsy's a singer, so he wrote this song. For those of you that aren't familiar with Happy Days, you might recognize that song just the same. Anybody know where else it's from? I mean, it's originally from the show.
It sounds familiar.
Yeah. It was also used as the St. Joseph's aspirin commercial for a while. So if you go on YouTube and you look up St. Joseph's aspirin commercial, you'll probably find it.
So we could make a video for you that you think the whole class could sing.
That's a possibility,
That
sure.
would be
Anyways, so it's actually a pretty good song to hit all the different parts. It's a little silly, of course, but it does sort of go through all of the circulation steps, and you can follow along. If you go to YouTube and you look up Pump Your Blood, that's the name of this song, there are versions of it where it has the lyrics on the screen if you want to try to follow along a little bit better. So there you go. Okay, so next week we'll continue on looking at the hearts. I'll have the models out at the beginning, and then we'll look at the physiology, which includes the EKG stuff, and we'll do all that. And hopefully we'll get to the blood vessels too. Question?
I was just wondering, just clarifying that there are no due dates, because on Blackboard it was saying it was like a late assignment that wouldn't take my newest score.
I went through right before class and made sure everything that was counted as late was checked off. I have to actually go through and look at the due dates for, are you talking about blood? Or
Just in general,
oh.
like any of those assignments, like the 1, 2, 3, and part 4.
For the cardiovascular assignments, I know, because this is, like I said, last summer instead of last semesters, I know the due dates are screwed up. Like there aren't due dates for some of the stuff. So I have to go through and do that for all the cardiovascular stuff anyways.
The due dates was messed up for Unit 3 as well too, because it was saying all of it was due like June 23rd
Until
instead of last night.
I thought I fixed that.
Nope, it was
Okay,
saying
well,
the 23rd.
I'll make sure things are
So
correct.
Unit 4 is due next Tuesday night.
Say that again?
Unit 4 is due next Tuesday.
Not all of Unit 4, just the blood chapter stuff.
Okay.
Because we're going to continue Unit 4 next week with the heart and the vasculature. So just the blood stuff is due next week. So have a nice week.
So now I can continue